Imagine your brain as a bustling city where neurons are the busy citizens constantly chatting and exchanging messages. At the heart of this communication are tiny messengers called neurotransmitters. They're the emails or texts that keep everything running smoothly. Now, one of the most fascinating messengers is acetylcholine, which acts like a friendly courier delivering signals to specific spots called receptors. Some of these receptors are muscarinic acetylcholine receptors, specialized locks on the neuron surface that only certain keys can open, like acetylcholine. These are part of a family called metabolic receptors, which don't just open channels directly, but set off a cascade of internal signals inside the cell, kind of like a domino effect. When acetylcholine binds to these receptors, it triggers a series of events that influence how neurons behave. Picture a neuron as a busy train station with synaptic vesicles, tiny cargo containers filled with neurotransmitters, ready to release their content when the time is right. When a neuron is activated, these vesicles fuse with the membrane at the synaptic terminal, releasing their neurotransmitter cargo into the synaptic cleft, the tiny gap between neurons. In the hippocampus, a brain region crucial for memory, neurons form intricate connections called synapses. Here, the post-synaptic side is packed with a dense zone known as the post-synaptic density, a kind of bustling scaffold of proteins that organize and support the signaling machinery. Think of it as a well-organized workshop where all the tools and parts are neatly arranged for efficient communication. This PSD is a proteomic marvel, composed of hundreds of different protons working together in multi-proton complexes. These include membrane receptors like NMDA and AMPA, glutamate receptors, channels that allow ions to flow in and out, and scaffolding proteins like PSD95 that hold everything in place. The clustering of these receptors at postsynaptic sites is vital for synaptic strength and plasticity, the brain's ability to adapt and learn. Diving deeper, you'll find signaling proteins such as kinases and phosphatases. Think of them as the switches and regulators that turn signals on and off. For example, calcium calmodulin-dependent protein kinase 2 is activated by calcium ions entering through NMDA receptors, leading to changes inside the neurons that strengthen the synapse, crucial for learning and memory. Within these complexes, GTPases and regulated proteins modulate the activity of signaling pathways, while cytoskeleton proteins provide structural support, shaping dendritic spines, the tiny protrusions on neurons where synapses form. The dynamic remodeling of these spines underlies synaptic plasticity, allowing the brain to rewire itself in response to experience. Disruptions in these synaptic processes can lead to neurological conditions like schizophrenia, where dendritic spine defects and aberrant synaptic plasticity cause cognitive and behavioral changes. So next time you think about brain communication, remember to fine-tune orchestra of receptors, scaffolds, and signaling pathways and metabolic influences, all working together in a symphony of life, learning, and adaptation.